Hello everyone, myself Rahul Kumar from Indian Agriculture Research Institute, New Delhi, and I welcome all of you in the virtual international conference on advances in food, agricultural science and technology 2022. And the topic of my presentation is mapping of QTLs for yield related traits in soybean. So as you all know, the most significant goal in the soybean breeding is to increase the seed production and overall yield of the soybean. And the expression of the various plant growth components such as plant height, days to blooming, days to maturity, number of pods per plant, pod length, pod width, and seed size features ultimately contribute in the seed yield, and they all are complicated quantitative traits that are majorly influenced by the environment. And therefore, there's a need to identify the chromosomal regions which contain the genes that regulate the yield-related traits in soybean, which can be achieved through the molecular markers. So therefore, we have started our research to investigate the inheritance and identification of QTLs controlling the yield-related traits in soybean. And for that, a cross was made between AGS 457, which is a bold-seeded variety of soybean, and a grain type soybean SKF 148, which is a small-seeded variety. So in this figure, you can see in the left side, it is AGS 457 variety, which is the bold seeded variety and brown seed coat color. And in right side, it is SKF 148, which is a small seeded variety and yellow in color. So we crossed these two variety and we generated the F1s. From F1s, we generated the F2, and from F2, we generated the F3. Then we did the genotyping in the F2 population, and we did the phenotyping in F2 derived F3 population. So first of all, a parental polymorphism survey was conducted with 580 SSR markers, and out of that 104 markers were found polymorphic between these two parents, AGS457 and SKF148. And these polymorphic markers were further used for genotyping the F2 population, and we created the linkage map. And after that, F2 and F2 to F3 population were used for the phenotyping. And by combining the genotypic and phenotypic data, we conducted the QTL experiment by using the software QTL ICI mapping version 4.0. So by using the genotypic data, we constructed the linkage map. And here you can see in this figure, using that 104 SSR markers, we created the linkage map for all these 20 chromosomes. And coming to the result, we can see we studied the all yield related traits such as plant height, seed weight per plant, two seed pod length, two seed pod width, number of seeds per plant, and seed weight per plant. And we found that for all the traits, uh, they show the normal distribution, which shows that these traits are the quantitative traits and governed by the large number of genes. So by using the genotypic and phenotypic data, we identified the QTLs for these traits. And you can see for the plant height, we identified 4 QTLs on chromosome 1, 4, 11, and 18. And out of these, we identified three major QTLs on chromosome 4, 11, and 18 with the R square value of 19.68, 17.07, and 10.09%. And for the number of pods per plant, we identified 2 QTLs on chromosome 20. And both these QTLs were the significant QTLs, explaining the R square value of 23.14% and 17.12% respectively. For two seed pod length, we identified 4 QTLs on chromosome 4, 7, 13, and 16. And here also we identified the major QTLs, which you can see in the R square values. And for two seed pod width, we identified 2 QTLs on chromosome 3 and 10. And here, one major QTL was identified on chromosome 10 with a R square value of 12.86%. For number of seeds per plant, we identified 2 QTLs on chromosome 16 and 20. And both these QTLs were the major QTLs. And here you can see the R square value, which is 19.37% and 18.67% respectively. And for our last trait, that is the seed weight per plant, we identified maximum number of QTLs. So for this trait, 5 QTLs were detected on chromosome 6, 11, 12, and 13. And you can see the position in the centimorgan and the marker interval for these QTLs. And out of these 5 QTLs, we identified 3 major QTLs on chromosome 11, 12, and 13. And this is one of the very important trait which ultimately determine the overall seed yield in the soybean. So the QTLs which are reported for this trait, the major QTLs can be used in the breeding program. So in conclusion, a total of 19 QTLs were identified for all the yield-related traits, with a single QTL accounting between 3.27 and 23.14% of the phenotypic variation. And these QTLs were found on 12 different chromosomes, and maximum number of QTLs were discovered on chromosome 20, which was 3 in number. And if I talk about the trait, then the maximum number of QTLs were identified for seed weight per plant, which was 5 in number, and 3 of that were the major QTLs. So in this current study, we identified 11 novel QTLs for the related traits, and our sum of the identified QTLs were the consistent with the previously reported QTLs. So the future prospect of this study could be the fine mapping of the reported QTLs, because in our reports the confidence interval is bit high. So the fine mapping of the major QTLs can be performed, and ultimately that major QTLs can be used for the introgression in the soybean genotypes in the breeding program for the genetic improvement of the soybean variety. Thank you.